Good afternoon, students. I will start the lecture in a minute. Please wait for a minute. Good afternoon all students. I'm going to start the lecture, and today's topic of our discussion will be Archaebacteria. Okay, will be the Archaebacteria. Okay, so what are Archaebacteria? Okay, simply if you want just to write one single line, okay, if you want to just write one single line about Archaebacteria, you can write simply they are extremophilic. They are extremophilic organisms. Okay, Archaebacteria are basically extremophilic organisms. Okay, so what are the properties? What are the general properties? You must have heard some of the properties. They can grow in the extreme conditions. In this unit, we are just going to discuss some of the properties of some of the archaea. Okay, which are specified in your syllabus. And the Archaebacteria, starting with a small video of the Archaebacteria from the National Geographic. Okay, starting with the video from a National Geographic.
Intense heat, freezing cold, high acidity, and radioactivity. These harsh environments don't seem hospitable for life, but some organisms not only survive, but thrive under such extreme conditions. The name extremophile means extreme lover. These organisms live in exceptionally harsh environments, such as hot hydrothermal vents or buried in rocks far beneath the Earth's surface. Extremophiles occur in all three domains of life: bacteria, archaea, and eukaryote. They range from the extreme heat-loving thermophiles, which feed off of inorganic chemicals and have special enzymes to survive high temperatures, to extreme cold-loving psychrophiles, which have evolved antifreeze proteins that help ensure their survival in some of the coldest waters on the planet. But the most extreme living things on Earth are tardigrades. Also known as water bears, these water-dwelling microanimals are polyextremophiles. This means they are capable of surviving multiple harsh conditions. They are nearly indestructible and have even survived the extreme conditions of outer space. Tardigrades have a unique adaptation that allows them to curl up into a dry, seemingly lifeless ball and slow down their metabolic rate. In this state, they can survive cold, dry environments like space for decades. Studying the adaptations of extremophiles may hold the key to solving many of Earth's problems. This includes the development of genetically based medications, producing new types of biofuels, and protecting people against radiation exposure. Extremophiles have opened our minds to the many possibilities of life and the environments that can support it. Understanding the limits of life in these extreme conditions on Earth may provide scientists with clues of how life could possibly exist elsewhere in the universe.
Okay, moving forward with the lecture. Okay, this was a video from the National Geographic. And basically the points which we are going to cover in today's lecture are basically general characters of the archaea, and types of extremophilic organisms, in which we are going to study overview of thermophiles, halophiles, acidophiles, alkalophiles, barophiles, as well as the methanogens. Okay, so starting with the archaebacteria. Archaebacteria, okay, archaeos means ancient, and a bacterium means a small rod. And is a unique group which is diverse in its morphology and physiology from the eubacteria and eukaryote. Okay, and as I told you, they are found in the extreme environments, okay, like highly saline or with high temperature conditions, or extreme cold conditions, or extreme pressure conditions, etc. Even with a higher irradiation, which includes hot springs, salt lakes, even the Antarctic desert soils. And they may be gram negative or gram positive, may be spherical, may be lobed, may be rod, spiral, or plate like, or maybe pleomorphic also. And their size ranges from 0.1 to 15 micrometer, and they can multiply by the binary fission. Okay, so basically the domain archaea is basically a diverse domain similar to that of bacteria. Okay, but this domain contains mostly are basically extremophiles. Okay, and some archaea are even mesophiles, and most of them are extremophiles, which may prefer hot or cold or saline or other conditions that are hostile to most other forms of the life. And their metabolism is basically adapted to the harsh environments, and they can perform even methanogenesis. Okay, whereas the methanogenesis is not performed by any of the eukaryotes or even the domain bacteria. Okay, and the size and the complexity of the archaeal genome makes it difficult to classify also. So they have been currently divided into 5 major phyla. Okay, they are divided into 5 major phyla, starting with Criniarchaeota, Euarchaeota, Coraarchaeota, Nanoarchaeota, and Thaumaarchaeota. Okay, and these are likely many other archaeal groups that have not been systematically studied and classified. And basically on the basis of phenotypically, the two main groups, major phyla, okay, two major phyla, Criniarchaeota and Euarchaeota. Okay, whereas basically Euarchaeota includes Methanogens and extreme Halophiles. Okay, and Criniarchaeota basically contains sulfur metabolizing Thermophiles. Okay, now moving forward with the general properties of the archaea. Okay, nutritionally they are either aerobic or maybe facultative anaerobic or maybe obligatory anaerobic also, and they may have a chemolithotrophic or to even organotrophic, or even some can also fix carbon dioxide, it means autotrophy, or some can even fix carbon dioxide by reverse TCA cycle or by reductive acyl CoA leading pathway leading to the production of the glucose via pyruvate also. And the plasma membrane of the archaea is somewhat different from that of eubacteria and the eukaryotes in having branch change hydrocarbon attached to the glycerol connected by the ether links. But in the case of bacteria, it basically fatty acids are connected by the ester links, whereas in this case they are connected with the ether linkage. The Archaebacteria chromosomes, okay, have a single circular DNA as in the case of eubacteria, but in a smaller length, and the archaebacterial TYC arm, okay, of the tRNA lacks thymine. You must have studied tRNA, which contains pseudouridine or 1-methyl pseudouridine. And the first amino acids to initiate a polypeptide chain is the methionine in the Archaebacteria, whereas in the case of eubacteria it's N-formyl methionine. It is N-formyl methionine. This is the basic difference between archaea and the bacteria. And this basically eubacteria are sensitive to chloramphenicol, but archaea are not sensitive to the chloramphenicol, while the diphtheria toxin have effects on archaebacteria but not on the eubacteria, and they have a very high variation, okay, in the GC content, okay, from 21 to 68% of the molecular weight of the molecular weight, okay. And the archaebacterial may have plasmids also, and the archaebacterial mRNA are more similar to that of eubacteria than to that of eukaryotic mRNA. What are plasmids? I forgot to tell you. Plasmids are just a circular form of DNA which are basically extra chromosomal material other than the main DNA, other than the parental DNA, okay. And these plasmids are basically carriers of information which are specific to a bacteria, which can be also transferred to the other bacteria. And some methanogens, okay, some of them are methanogens. In the case of archaebacteria, have histone like proteins that binds with the DNA to form a nucleosome like the structure. And the speciality of the methanogens is the presence of the coenzymes F420M for 30 methanofuran, methanoptene, and coenzyme M. Okay, you will study structures in your semester 5. The extreme halophiles are only photosynthetic archaebacteria and convert light energy into chemical energy by means of a proton pump based on a pigment rhodopsin. What is bacteriorhodopsin? Okay, uh, similar to that of bacteriochlorophyll, but the structure is different. The function is similar to that of a bacteriochlorophyll, but the structure is different. Its structure resembles to more of a retina, okay, to the more of a retinal one. Now the group of the archaea, which includes methanogens, cellulose archaea, sulfate reducing archaebacteria, haloarchaebacteria, and the extremely thermophilic bacteria and the extremely thermophilic archaebacteria, okay, where sulfate also includes acidophiles also, and halophilic also includes alkalophiles also. Now starting with the methanogens, As the name itself says, methanogens are basically the bacteria which are able to produce methane, okay, which are able to produce methane, okay. And the methanogens, other than the different organisms, okay, they are a diverse, a phenotypic and a ecological group which is basically divided into methanococci, methanobacteria, and even methanomicrobia, okay. And they may be gram negative or gram positive with a wide range of morphological types, cells or coccoid bodies, and pseudosarcina or rods. They may be motile or non-motile and are strictly anaerobic, and its main specialty is they produce methane. And this methanogens require specialized conditions also, which methanogens, which are also called methanomicrobials, form a highly specialized physiological group which do not utilize carbohydrate, proteinaceous materials or other organic compounds other than there are listed, and they grow by oxidation of hydrogen or formate with the reduction of the carbon dioxide to methane, okay, and by the fermentation of methylated amines, methanol or acetate to methane, carbon to methane and carbon dioxide, okay. And some strains can even oxidize 1-propanol, 2-propanol, 2-butanol, ethanol to the propionate, acetone. Next one is 2-butanone or acetate, and reduce carbon dioxide to the methane. And the some strains reduce elemental sulfur also, but this may not lead to the growth as well. Unique properties: they do not contain muramic acid. Cells lipids are predominantly isopyramid hydrocarbons which are ether linked to the glycerol, and cell contains coenzyme M and coenzyme F420 and they are widely distributed in nature and are being found in the anaerobic habitats such as aquatic sediments, anaerobic sewage or sludge digesters, and the gastrointestinal tracts of the animals, okay. Here are some of the examples of the methanogens, okay. I'm just going to read all these: methanobacterium, methanobrevibacter, methanospira, methanothermus, methanococcus, methanocorpusculum, methanosulilius, methanogeum, methanoleucinium panteri, methanomicrobium mobile, methanoplanus, methanospilum huncti, methanococcus, methyllutens, Methanohalobium avigatus, Methanolobus, Methanosarcina, and the last one is Methanothrix. Okay, I have included one of the example from this group, which is an two examples: Methanobacterium, Methanococcus. Okay, gram results are basically variable and may vary from species to species, and they are crooked to straight rods along to the filamentous and about 0.5 to 1 micrometer in width. Motility: they are non-motile endospore, they are not endospore formers but do produce fimbriae, okay, and are strictly anaerobic Optimum temperature required for their growth is around 37 to 45 degrees Celsius for the species which are mesophilic, and for the thermophilic species they require about 55 degrees Celsius. Okay, you must have also heard of the biogas plant, okay, which is used for the production of the methane. In that conditions, basically thermophilic methanogens are the one which are used for the production of the methane, and methane is produced with a temperature range from 50 to 55 degrees Celsius. Okay, now moving forward with the requirements, ammonia can be used as a sole nitrogen source, and sulfide may serve as an sulfur source. Energy metabolism by reduction of carbon dioxide to the methane. Okay, and electron donors are limited to hydrogen, formate, and carbon monoxide And the second X, I told you I have taken used two examples. Second one is Methanococcus. Cells lose integrity during gram staining and lives within 10 seconds. And when suspended in distilled water or 0.051 normal sodium dodecyl sulfate. Okay, so gram staining, it's a very hard task while staining of Methanococcus. And the morphology, they are irregular coccus with a diameter of 1 to 2 micrometer during the balanced growth, and in the case of extreme conditions or in the older cultures, the shapes of the cultures and colonies are also irregular. They are motile by means of polar tufts of the flagella. Okay, we have already studied what do you mean by polar tufts of flagella. We have already studied. And the storage materials include glycogen. Tropism, with one exception, all species grow autotrophically in the mineral medium. They are obligatory anaerobic and have a pH optima between 6 to 8 and a temperature they may be either mesophilic with a temperature optima of 35 to 40 degrees Celsius. If any they are if they are thermophilic, their optimum may be around 65 degrees Celsius. If they are extremely thermophilic, then the temperature range would be around 85 degrees Celsius. The amino acids and acetate are stimulatory for some species. The nitrogen sources include ammonia, nitrogen gas, as well as the alanine. Okay, and other unique features are they require sodium chloride for the growth and with an optimal concentration ranging from 0.5 to 4 percentage. Okay, and obligate methanogens basically use hydrogen. Okay, and in the most cases format also serves as an electron donors. In this case, acetate, methanol and methylamines are not the substrate for the methanogenesis. They are found in salt marsh conditions, marine environments, as well as the estuaries. Okay, as well as the estuaries. Please have a look at the Methanococcus. Another example, which is basically of a methanomicrobia, can make a methane from the acetate or methanol rather than the carbon dioxide also. And most of the methanogens are basically autotrophs and use a methanogenic pathway for the carbon dioxide fixation as well as the energy production. Okay, while some of we also use acetic acid, okay, for the production of methane also. Okay, and the enzymes basically which are used in methanogenic pathway are extremely oxygen sensitive, and so all the methanogens are extreme anaerobes. However, there are organisms found in all anaerobic environments and are certainly the most prevalent and the cultivable archaea in the moderate world also. Okay, there are some examples of them also. Okay, please right now please have a look of other methanogens which are Methanobrevibacter smithi. Okay, this is an scanning electron microscopy photograph, and the other one is an Methanococcus bartoni, which is an TEM photograph. Okay, and the examples which I told you, which are also found in the moderate world. In the case of sediments and soils, a swamp gas is basically methane. Okay, and because of its low ignition temperature and the threshold concentration, is basically readily ignited and even glows very faintly as a will of whips which are may be visible in the night, okay, which may be visible in the night in the swamps also. And the methanogens are crucial components of the microbial populations, okay, of the rhizosphere. And other scenarios include animal gastrointestinal tracts, okay, animal gastrointestinal tracts, especially there are some examples of wood eating insects and the ruminants, and but most other animals as well, okay, may have methanogens. And one such example of an insect is an African termite mound, okay, and another example is a cow, okay, where cow may produce enough methane to be a, okay, and they are also found in the wastewater and the landfills, okay, and the landfills and the wastewater process basically converts organics into wastewater into methane and carbon dioxide. And landfills, what are landfills? Landfills are basically structures where all the solid waste is being dumped, okay, and landfills must be carefully vented and housed near the old unvented landfills, okay, may lead to even explosion in such of the landfills also, okay. And you all know the oil deposits, okay, you already know the oil deposits which may contain the natural gas, which may contain natural gas methane. Okay, now moving with the next group, Salvol-less archaea. Salvol-less archaea, okay, they stain negatively in the Gram reaction and have a coccoid cell, okay, may be motile and flagellated. Coccoid cells like a cell envelope, facultatively anaerobic, obligate acidophile, okay. So these are acidophiles, obligate thermophile which grows at 33 to 67 degrees Celsius and require yeast extract for their growth. And the cytoplasmic membrane contains mannose rich glycoproteins and a lipoglycan also. They are found in sulfating coal piles and acidic sulfophiles. Okay, so they are basically found in the coal mines. Okay, which includes this includes genus Thermoplasma. Okay, and the species is Thermoplasma acidophyllum and Thermoplasma volcanicum. Okay, now the Thermoplasma. So what are thermoplasmas? Okay, and what are the basic properties of the thermoplasmas? Okay, now thermoplasmas are basically chemoorganotropes, okay, and are strictly aerobes, but some of them are also facultative, some of them are even facultative anaerobes also, and they are obligate thermoacidophiles with a pH optima around 1 to 2, and the cells undergo lysis near the neutrality, and the obligate thermoacidophile, okay, requires about 55 to 59 degrees Celsius for their growth, and may even use sulfur anaerobically for the respiration, okay, even use sulfur for the respiration. Now this one here is an photograph of a thermoplasma, okay. Now the thermoplasma acidophilium as the name suggests, it's an acidophilic. Okay, and their shape is irregular. Okay, their shape is basically irregular. Okay, with the cytoplasmic extensions similar to that of pseudopods. Okay, you can clearly see the shape here in the photograph given here. Okay, which may have similar look like the amoeba. Okay, which are basically eukaryotes. Okay, which are basically eukaryotes. Okay, and the cross linking of basically carbohydrate chains of membrane glycoprotein provides cell rigidity and the osmotic tolerance, and the osmotic tolerance in the case of Sulfolabus archaea. Okay, now moving forward with the next group, which includes sulfate reducing archaebacteria, blue green fluorescence of 420 nanometer under the UV microscope, and are strictly anaerobic and are strictly anaerobic. Okay, they are extremely thermophilic and reduce sulfate to form hydrogen sulfide, and even produces trace amount of methane, and RNA polymerases is of A plus C and B type, and cell processes factor F420 and methanoptene, but the coenzyme M and cofactor 430 is not present, where in the case of methanogens all these factors are present for the production of methane. Okay, the examples are Archaeoglobus fulgidus and Archaeoglobus profundus. Okay, now moving forward with the Halophilic archaea. Okay, before moving towards the Halophilic archaea, let me give you some information regarding this bacteria also. Archaeoglobus fulgidus. Okay, basically they are found in a deep sea hydrothermal vents and the heated marine sediments, and it's a thermophile which grows at 85 degrees Celsius. Okay, and some of the species are motile also. Okay, and this bacteria can grow autotrophically by sulfate reduction using hydrogen as the electron donor. Okay, and sometimes it can grow heterotrophically also from the lactate or acetate, plucking the methyl group from this, in essence of using methanogenic pathway in reverse to generate hydrogen and carbon dioxide. Now halophiles, as the name itself suggests, okay, they are basically salt loving organisms. Okay, a diagram given here on the left side is basically of a salton in Australia. Okay, where you can see the square shaped cells as well as cocci. Okay, which are basically halophiles, and on the right side given here B diagram is basically a salt evaporation pond which is extremely high in salt and mineral content. Okay, and the archaea dominate these hot and saline habitats, produce red pigments. Okay, now this one is a photograph of a Archaeoglobus. Now this one is a photograph of a methanotrophic archaea which grow in association with the sulfate reducing bacteria. Okay, and the cluster of methanotrophic archaea stained red by an archaeal fluorescent 16S rRNA probe, and which is surrounded by a sulfur reducing bacteria which are labeled by green 16S rRNA fluorescence probe. Now halophilic archaea, the cells are maybe gram negative or gram positive, rods and regular to highly irregular cells also. They are basically chemo-organotrophs. They may be aerobic or facultative anaerobes, may be neutrophilic to even alkalophilic. Okay, they may be mesophilic or slightly thermophilic also. Okay, but the main condition is they require higher concentrations. They require higher concentrations of the sodium chloride for their growth. Okay, and some of the species contain bacteriorhodopsin which are able to use light for the ATP synthesis. Okay, when we study the photosynthesis, okay, I tell you the bacterial chlorophyll is the pigment utilized for the photosynthesis, which is utilized for the photosynthesis. But in this case also some of the species contain bacteriorhodopsin, okay, which work as a pigment for the generation of ATP synthesis, and some of the species even contain carotenoids which give a coloration according to the type of carotenoids present in the halophiles. Now the examples are Haloarcula, Halobacterium, Halococcus, Halophylex, Natanobacterium and Methanococcus oculatus. Methanococcus oculatus. Okay, the Halococcus, one of the example which I have used here is of a Halococcus. They are mainly gram negative. Some of them are gram positive. Okay, they are cocci shaped and have a diameter of around 0.8 to 1.5 micrometer in diameter. Occurs in pairs, tetrads, sarcina, packets, or even the irregular clusters. They are non-motile and divide by the septation, and colonies are red to orange. Due to the carotenoids. Okay, as I told you earlier that the halophiles may contain carotenoids. Okay, according to the basis of which carotenoids is present, they may be present in a different types of colorations. They are chemo-organotrophic, they are strictly aerobic and have a optimum temperature around 30 to 37 degrees Celsius and require at least 2.5 molar of NaCl for growth, and but the best growth occurs in a salt concentration with a range of 3.5 to 4.5 molar NaCl. Okay, they are oxidase and catalase positive. Okay, this one is in photograph of an Halococcus. Again, this one again it's an photograph of an Halococcus. Basically, the habitat of the Halophiles, okay, they are generally found in hypersaline seas, lakes, salt evaporation pools, salted meats, even salt marshes, or even in the salt deposits. And they can be also found in unexpected low water containing environments such as soil and even the sludges. Okay, and what is his specialty behind this mechanism to survive? Okay, most of the haloarchaea, okay, when there is little net osmotic pressure on the cell wall, okay, that is basically high salt concentration both inside and out. uh, some species basically take advantage to this. So by adopting a high surface flattened area shapes like which you have seen in the photographs, cube shaped, okay, triangle shaped, okay, they are not possible which are basically not possible for normal organisms with the regular cell process. Okay, and there are also some halophiles which grow at high pH. Okay, where proton pumped are basically pumped outside by the electron transport or by the bacteria rhodopsin also, which basically reacts with the hydroxide in the environment very quickly also, and some of the protons which are present at very low concentrations outside the cell to drive the ATPase also. Also, the electric potential is still present. It cannot be harvested by ATPase unless enzyme gets protons from the outside. In any of the cases, cells need protons back to maintain the inner pH of the cytoplasm also. Okay, so by this problem actually limits the maximum pH compatible with the life also. Okay, now the next group, which includes extremely thermophilic archaebacteria. Extremely thermophilic archaebacteria, which includes acidenius, Sulfolabus, Sulfolabus, ambivalence, hyperthermus, butylicus, Metallospira sedula, Pyrobaculum, Pyrococcus, Pyrodictum. Okay, and Staphylothermus marinus, Sulfolabus, Thermococcus, Thermodiscus, Maritimus, and Thermophilium and Thermoproteus also. And one of the example which I have used is of a Sulfolabus. Cells are coccoid, highly irregular also, and have a diameter of 0.8 to 2 micrometer in diameter, and usually occur in singles. They are not motile, and the cells envelope are composed of protein subunits in the hexagonal arrays. The unique features are the presence of isopronyl ether lipids, calditol, Caladerelia quinone are also present. Okay, they may be lithotrophic, organotrophic, growth by oxidizing organic materials such as yeast extract, sugars, or even amino acids. Aerobic growth via oxidation of sulfur, sulfite, tetrathionate, and no anaerobic growth via sulfur reduction. They are thermoacidophilic, which grows at a pH of 1 to 6, okay, and grows at above 50 degrees Celsius, okay, and grows above 50 degrees Celsius. Okay, please have a look of a photograph of a sulfobus. Now next one is a sulfonema. The next one is a sulfonema. The gram stain may be positive and sometimes unequally stained, but in the electron microscopy, ultra thin sections exhibit the cellular characteristics of a gram negative bacteria. Okay, please note this point properly. The cells arrange in uniseriately multicellular filaments are 3 to 8 micrometer in diameter and sometimes 1 millimeter in length, and the nearly cylindrical cells are 2.5 to 13 micrometer long, and the cross walls are sometimes also visible. They have flexible filaments with the gliding motilities, and the granules of polybeta hydroxybutyric acid are commonly stored, and the filaments are always attached to the surfaces that are necessary as strap data for the gliding moving, and the outer membrane has a wavy structure, okay. Next one, disulfonema again. The gliding movement and the growth are promoted by addition of insoluble substrata, okay, such as agar, or inorganic precipitates, They may be chemoorganotropes or chemolithotropes, okay. They are strictly anaerobic, can follow a metabolism. Respiratory growth by fermentation is not observed, okay. And sulfates and other sulfur compounds serves as electron acceptors and are reduced to the hydrogen sulfide with a temperature range of 10 to 36 degrees Celsius, okay. In the anoxic media, containing reductant and vitamins are basically necessary for the growth, okay, and do require brackish water or seawater concentrations of NaCl and MgCl2, and in some cases also calcium chlorides, and cytochromes are basically present, okay. Please have a look at the D-sulfonema. Next one is Thermotoga. Next one is an Thermotoga. They are basically gram negative rods and are surrounded by sheath like structures ballooning over the ends of the cell, and the non-motile or motile bipolar, lateral or peritracheous flagella. Spores are basically not present. They are chemoorganotropes and they are anaerobes, okay. Temperature requirement, their optimum temperature for the growth is 70 to 80 degrees Celsius, but it has been observed they can also grow as high as 90 degrees Celsius, and the hydrogen basically is inhibitory, and the inhibition is overcome by the addition of sulfur with the formation of hydrogen sulfide, and they basically metabolize carbohydrates, tryptones, yeast extracts. The fermentation products are mainly lactates, acetates, carbon dioxides, and hydrogens, okay. And the main strains will grow in 3.75% NaCls and have been isolated geothermally in the heated marine sediments or the hot springs. Please have a look at Thermotogoa, okay. I'm going to end my lecture, okay. I'm going to end my lecture now, okay. And please join the WebEx session for the discussion. Please join the WebEx session for the discussion, okay. I'm ending my stream.